A previous video on mutations was kind of cut short, so I wanted to do a little addendum to the end to make sure that you understood the idea of gene mutations. Their mutations can happen at many different levels. They can happen at the chromosomal level, where entire genes are duplicated or deleted or inverted. That was part of the previous video on mutations. But here's a little more smaller scale at the nucleotide level. You can still have detrimental effects. These are called into two categories as point mutations. And point mutations generally will affect one point or one nucleotide in the sequence. So we have a substitution, and they often introduce this by taking it out of science into nucleotide language, because that might confuse some students, to putting it into three-letter words. So the fat cat ate the rat. If you notice, the reason why they do everything in threes is because this relates back to protein synthesis with codons. In the end, mutations are going to affect the proteins that are affected or made and put together by the ribosome. And the ribosome reads everything in codons, and codons are 3 nucleotides at a time. So that's why you're going to see the commonality. It's not because they just want a simple sentence. It's 3 letters, and these 3 letters would correspond to an amino acid in the end. So the fat cat ate the rat was the original. A substitution is taking one of those letters and substituting it for something that doesn't stand there. It does make a new sentence in this example, but the fat hat ate the rat. It is a substitution error. It is a different word here, so that does have an issue, and there could be genetic diseases because of that, such as sickle cell anemia is the result of just one letter that's been substituted out and placed in for another nucleotide. Another category, they often put these into the Frameshift mutation category. It does affect one nucleotide. Here, the fat cat ate the rat transitions to having the addition of 3 letters, and that affects one amino acid in the end. I can still see the fat cat ate the rat. This actually is not as detrimental because when you incorporate just one extra codon, then we have one extra amino acid. Does it have an effect? Yes. As opposed to what would be called a Frameshift mutation, where they insert just one letter. This letter X might come in there, and all it would be is the fat cat, and then you'd see the X, A, T. That would be the next 3-letter word. It's not a word. And then it would shift everything down. So it would really become very confusing from the point of the insertion downstream to that. So you would never see ate the rat if you only had one letter. The body doesn't recognize just one, say, oh, this is a mistake, so I'm just going to read this one. I know that I have to read the next 3 together. That's not how it works. The deletion, for this example, they get rid of an entire sequence or codon. That doesn't always happen. You can just get rid of one letter. And so this would be the fat, atta, tet, her, at. Like if you can kind of follow that. If I take the letter C out, I've got to adjust my reading frame. That's why they call it a frame shift. So this is all just simple non-science examples, but here's a chart to kind of show you what would happen in the long term. If this was an actually thing happening in DNA. Here's the original sequence. All these 3-letter abbreviations are your amino acids. The normal DNA sequence right here will correspond to this correct sequence of amino acids. But if we have, the first one here is what's known as a silent mutation. And a silent mutation is when you substitute a letter, but look at the effect, it's still a leucine. Because some codons code for the same sequence, you lucked out. There was a mutation, that's why it's called a silent mutation. So second example is the silent. The third example is kind of what I just did with the fat cat ate the rat, or whatever that sentence was. This is sometimes also known as a missense mutation. So a point mutation, another word for a missense as well, because it just changes one amino acid. It started as leucine, but now it has changed to serine because of this change right here. That T-thymine transitioned to a cytosine, and that's going to change potentially the amino acid. In the silent mutation, it did not. Even though we had one change, we lucked out. Here, the change did correspond to a different amino acid. So that's why it's a substitution sometimes as well, is because you substitute a different amino acid into its place. The next one, if you notice, you'd have to kind of be trained to do this, and you'd have to recognize what the outcome would be here. But what they did is they changed a letter, and ultimately, instead of it coding for an amino acid, which it did in the previous, this actually transitions to a stop codon. So this is known as a nonsense mutation. So a nonsense prematurely stops the amino acid sequence, and that's how you would recognize it. It's still a point mutation because it changes one nucleotide, but the outcome is a stopped or precursor, like it prematurely stops the amino acids from forming. So instead of having a sequence that's this long, you have a sequence that's this long. So that's called a nonsense mutation. Okay. And then what you have here, you have a frameshift, which I talked about, but here is just one base pair deletion. So you can't see, you can kind of go right to where it should be. So where this letter is gone. So now the T, A, C slide into place. So the change occurs from the point of the deletion, then the next three letters. So T, A, C, C, T, A. Now they're read together, which is leucine. So you start to see how there is a downward effect from the point of the deletion throughout the rest of the sequence. So here we go. Normal or typical sequence is what it should be. This is a silent mutation, a missense mutation, a nonsense mutation, or a frameshift mutation. And frameshifts could be a deletion or they could be an insertion. So they don't have an example of that, but you can kind of guess if you added a letter here, you'd have to read that in as still a 3-letter sequence. Okay? Hopefully that clarifies the last portion there of our gene mutations.